Hello, welcome to all of you in the hall and you on the stream. Introduction will be partly Norwegian and partly English, and both lectures will be in English. Vi starter nå 14. sesong med NTNU Kveld med presentasjonen av Kavli-prisene som tildeles i nevrovitenskap for årene 2020 og 2022. Mitt navn er Randi Wenche Haugen, formidlingssjef ved Vitenskapsmuseet på NTNU og prosjektleder for NTNU Kveld. Nyhetsbrev får dere ved å gå inn på ntnu.no/kveld og trykke på nyhetsbrev, så får dere informasjon om alle arrangement. Vi gjør TV-opptak i kveld, det legges på YouTube senere, og det er fotografering. Nødutganger, emergency exits, that's the back door, the side door and crossing the stage. Tonight, the Kavli prizes are awarded in Oslo. We will give you a presentation of the Kavli prizes awarded in Neuroscience. Due to the pandemic, the prizes were not awarded in 2020. Who won the Kavli prizes in Neuroscience and why? Tomorrow evening, we will do the same about the Kavli prizes in Nanoscience. Tomorrow evening, we will do the same about the Kavli prizes in Nanoscience at the same place here at the same time. Vi har 2 foredragsholdere i kveld. Før pause får vi høre om vinnerne i 2020, og etter pause får vi høre hvem som vant i 2022, og hva de har funnet av ny kunnskap. Kort pause 15 minutter, og etter hvert foredrag kan det stilles spørsmål. Hva er Kavli-prisen? Kavli-prisen ble opprettet av norske Fred Kavli for å fremme vitenskapen til fordel for menneskeheten. Fred Kavli vokste opp på 1930-tallet blant mektige fjelltopper i Eresfjord i Romsdal. Han var nysgjerrig og utforskende, og etter NTH i Trondheim så reiste han til USA, hvor han gjorde stor suksess med et sensorteknologi for fly- og romfartsindustri. Basert på denne formuen stiftet han The Kavli Foundation og The Kavli Prize, som deles ut annethvert år i 3 vitenskaper. Astrofysikk, nanovitenskap og nevrovitenskap, som er det største, det minste og det mest komplekse. Fred Kavli lå ofte som guttunge utendørs i nattemørket og undret seg mens han stirret opp på stjernehimmelen og det uendelige universets store mysterier. Han mente disse 3 var de mest spennende forskningsfeltene for det 21. århundre og utover. The Kavli Prizes was established by Norwegian Fred Kavli to promote science for the benefit of humanity. He grew up in Norway in the 1930s among mighty mountain peaks in Eresfjord in Romsdal. He was curious and exploratory, and after NTH in Trondheim, he traveled to the USA, where he was very successful with sensor technology for aircraft and the aerospace industry. Based on his fortune, he founded the Kavli Foundation and the Kavli Prizes, which are awarded every 2nd year in 3 sciences: astrophysics, nanoscience, and neuroscience, the biggest, the smallest, and the most complex. As a boy, Fred Kavli often lay outside in the dark night and wondered as he looked at the starry sky and the great mysteries of the infinite universe. He believed these 3 were the most exciting fields for research in the 21st century and beyond. Vinnerne av Kavli-prisen i nevrovitenskap i 2020 fikk de prisen for oppdagelsen av reseptorer for temperatur og trykk. The winners of the Kavli Prizes Neuroscience 2020 received the prize for the discovery of receptors for temperature and pressure. The lecture is given by Jonathan Whitlock, Associate Professor at the Kavli Institute for Systems Neuroscience. The stage is yours.
Thank you, Rondi. Good evening, everyone. Let me get my presentation queued up. You see it okay? Yep. Okay, so tonight I'll be presenting on the Kavli Prize in Neuroscience, the winners from 2020. Obviously, due to COVID-related restrictions, things weren't happening in person. Talks were given virtually and online. But the prize winners, along with members of the Kavli Foundation, will be in Trondheim. There will be all kinds of events and lectures and public forums that are going on this week. I have the date and time for I Meet the Speakers, a presentation that will happen later this week at the end of my talk. So as Rondi had just explained, the Kavli Prize is awarded every couple of years, every 2 years, to honor breakthroughs in the fields of astrophysics, nanoscience, and neuroscience, the big, small, and the complex. The prize consists of $1 million US for each of the scientific fields that are involved. So in sort of staging the air, if you will, for the topic of the prize, we can do a thought experiment. That's where you just imagine that you are on a sunny, warm beach, and you can feel the warm air of the beach blowing over your skin. You can feel the different qualities of sand under your feet. If the sand is coarse and grainy, or if it's very fine, you can feel the water lapping over your feet. And so these are all very familiar but quite distinct sensations, and they all depend on our sense of touch. So whether something feels hot or cold or watery, these are all part of the sense of touch, which has been one of the least well understood of the major senses until the work of the 2020 Kavli Prize winners, who are David Julius and Artem Pataputian. So David Julius is the chair of the Department of Physiology at University of California, San Francisco. And Artem Pataputian is a professor of neuroscience at the Scripps Research Institute in La Jolla and a Howard Hughes Medical Institute investigator. And they won the prize specifically for their transformative discovery of receptors for temperature and pressure. So what they did is a major breakthrough because the molecular basis for how temperature and pressure are encoded and transformed into electrical signals in the body has been lacking. So the two prize laureates discovered these receptors specifically for temperature and pressure. These findings revolutionized this subfield of sensory neuroscience because it provided a molecular neural basis for thermosensation, which is the sensation of heat, and mechanosensation, which is the sensation of mechanical touch. The topic of how we sense the world has been a matter of fascination for naturalists and philosophers for centuries and centuries and millennia. So Kant noted in his Critique of Pure Reason that all our knowledge begins with the senses, proceeds then to the understanding, and ends with reason. And in this drawing after René Descartes' Treatise of Man, from 1662, we see this drawing of a person who is standing or kneeling next to a noxious heat source of fire. And what Descartes reasoned was that this causes, whenever you feel something that's painful, it causes channels in the musculature to open so that animal spirits can then communicate from the outside world the sensory signals, then where it's ultimately experienced in the mind. So while we've made some progress since the 1600s on the biological basis of it, again, this has been a matter of fascination for quite some time. So not only are our senses the source of reason, they are totally fundamental for our survival. They ground us to the world. Our senses are the basis of all of our interactions that we have with the world and with each other in it. Our senses work through specialized sensors that are embedded in the tissues of our body, which interact with physical stimuli that arrive to us from the outside environment. The mechanisms for how we experience touch or temperature has remained a mystery, again, until the work of the 2020 Kavli Prize winners, which I should note also went on in 2021 to be awarded the Nobel Prize. So touch and temperature then are in contrast with senses like vision or smell, where we know much, much more about the mechanisms for how these other kinds of senses work. So I'll just describe those briefly. So again, sensation is what happens when some form of stimulus, if it's some form of energy or some sort of chemical odorant, arrives to our body from the outside world. So in the case of vision, photons stimulate cells, photons of light stimulate cells in the back of the retina. What those cells do is kind of remarkable. They convert the electromagnetic energy of the light, they convert it into a biochemical signal that then leads to electrical impulses that travel from the back of the eye, from the back of the retina, into the visual cortex. And how our visual system processes visual information was recognized. Those insights were recognized with the Nobel Prize in 1981. Another example is the sense of smell, where odorant molecules are sensed by receptors in the olfactory epithelium. This is a specialized kind of skin that is inside, behind your sinuses in your face. And these contain receptors that in a lock and key kind of mechanism will bind to an odorant molecule, and then this then triggers them to send electrical impulses, again, back into the brain. And the cellular basis for how we smell and how we detect odorants was again recognized with the Nobel Prize back in 2004. Then the sense of hearing, pressure waves in the air, they come at us from the environment. These cause your eardrum, the membrane in your ear, to vibrate. That communicates the waves back to a specialized organ, the organ of Corti, that sits in the middle of your ear, in your middle ear. And specialized hair cells in this organ shear, and when those hair cells shear, they send electrical impulses to the brain, and then this is the basis of hearing. And these biological mechanisms of hearing were recognized with the Kavli Prize in 2018. So in all of these cases, something is coming from the outside and being sensed by something very specialized and quite small to give you a certain type of sensory experience. So the senses of temperature and touch are different because these occur through direct mechanical contact that you have with something from the world. It's not coming from out there, it's actually on you. When something is touching you, it's actually touching the surface of your skin. And our touch sensors are not just confined to the eye or the ear, but they're all over the body. So it's everywhere, and it's not something that you can gate, and it's not something you can turn off, the way you can close your eyes or plug your nose. So if you take the example of touching a coffee mug, the coffee mug has different qualities to it. It's a smooth surface in this case. It's brittle, it's hard. If there is hot coffee inside of it, then you can also experience the temperature sensation in addition to the qualities of touch of the mug itself. And so if we consider, zooming in and looking at the fingertip a little bit more closely, we'll just stay here in the middle. Your sense of touch is amazing. Your ability to use your fingertips for tactile discrimination extends down to such fine scales that you can sense indentations 500 times smaller than a human hair. We rely on our sense of affective touch for our emotional health, for example, when we hug or embrace loved ones. We also use our sense of touch in the sense of proprioception, which is that we have stretch sensors that are embedded in our skin. And by virtue of this, you can close your eyes and you still have a sense of where your hand is located or how your body is configured in space. It's this kind of sixth sense that we have of body configuration that we kind of take for granted. But it's remarkable, and it is also possible through these mechanisms of touch. So then, if we consider the skin even more closely, you see that the skin consists of different layers. There are the deep layer, the middle layer, and the outside, the epidermis. This middle layer is called the dermis, and it's loaded with hundreds and hundreds of millions of individual receptors that sense different qualities of touch. So you can think of this as a touch receptor, a pressure receptor. There are vibration receptors that are buried a little bit deeper in the skin. There are receptors for warm and cold, and there are pain receptors here. And these are just bare nerve endings that extend from the dermis up into the epidermis. So what this illustrates is that our sense of touch is remarkably capable. It's incredibly fine. And there are different qualities to it. It's not just a unified sense, but there are different aspects of touch that these different types of receptors communicate. And these receptors, where I've been focusing, these are on the surface of the body. They're on the outside of the body, but these actually originate from nerve cells that live on either side of the spinal cord. This is actually where the sensory neurons in our bodies are located. The cell bodies are actually there. And what it is they send these tiny, thin little filaments out to all reaches of the body, all matters at the surface of the body. And it's at these very specialized endings that I was showing in the last slide where these different aspects of touch are sensed. So stimulus detections happens at sensory nerve endings, let's say out on the fingertip, and then they get communicated back up towards the cell bodies by the spinal cord. And so that's what's happening in the picture here. You have receptor nerve endings in your fingertip. They send signals, let's say from mechanical sensory input or for pain or temperature. These get communicated back to cells in this structure called the dorsal root ganglion. And every segment of your spinal cord has one of these ganglia on either side of it. The signals pass through the root ganglion and into the spinal cord, where the signals then are communicated back to your brain, where you consciously experience the sensation and you decide what to do about it. So on to the receptors themselves, the actual discovery that led to these breakthroughs and these prizes. So touch and heat receptors are embedded in these specialized sensory cells. So if you take this so-called pain cell from a couple of slides ago, you consider the free nerve endings that are reaching out into the dermis. Well, these cells contain different genes, and those genes express different proteins, and Miriam will be talking more about this. And so what happened in the lab of David Julius was they took different genes from these free nerve endings, and they expressed them in cells in a laboratory setting. And these cells normally don't feel, they don't sense anything. They're non-sensing cells. And the researchers painstakingly put one gene at a time into these cells and applied capsaicin, which is the active ingredient in chilies. And he screened one gene at a time until finally he found a gene that led to these otherwise non-sensing cells to go nuts and become very active whenever capsaicin was present. And then they identified the gene and found that it encoded a ligand-gated ion channel called the TRPV receptor. And this was the discovery. This is the thing that they found that was the big breakthrough. So this ion channel, as it turns out, it binds to the capsaicin molecule, and when it does that, it opens up. And when this ion channel opens up, ions can flow from outside of the nerve cell to the inside of it. And as these ions flow in, that causes the cell to become excited, and it sends electrical pulses back to the brain. And so this is sort of like a molecular flag or a sensor to say, hey, I've touched something, I'm active, and that thing happened to be capsaicin. So it was later found in subsequent experiments that if you just turn up the heat, if you simply heat the environment, or if you apply focal heat to the cells, that was sufficient to drive the receptors to become active as well. So there are different families of these so-called TRP receptors that sense all manners, different gradations of heat, from minor heat up to very, very hot heat. And the TRPV receptor, which is the original one that was discovered, is just one of this family. Another one that's quite interesting is the TRPM family. The M stands for menthol. And these receptors sense cold. And so what was found there is if you apply menthol to cells expressing the TRPM receptor, that the cells become active. And if you lower the temperature, that also causes the cell to become active. So if you take a bite of mint and you feel that kind of cool breeze as you inhale into your mouth, you feel that coolness on your tongue, that's literally the same receptor that goes off whenever you breathe in cold air. right? So from a sensory physiology perspective, it's the same thing, which is kind of remarkable. Then for the sense of fine touch, not temperature, but fine touch, there are different types of nerve cells that sense and transduce these aspects of touch. So the cells in which the piezo receptor was best characterized was the Merkel cell. And in this case, the researchers, they took cells that they knew were they took cell lines that they knew were very, very sensitive to mechanical perturbation. And they knocked out genes one at a time until they found a gene that made the cells insensitive to touch. And that's how the piezo receptor was discovered. They found a gene that when it was knocked out, the cells weren't active. And as it turns out, it was for this channel, the piezo-1 channel, that lives in the cellular membrane of these Merkel cells, among other cell types. And normally, when it's inactive, it's got this kind of bowl-type shape. It consists of 3 large leaflets that are put together. And whenever mechanical force was applied, not necessarily on the receptor, but just nearby it, that was enough to cause biophysical changes in the receptor so that the thing snaps open. And so mechanical touch causes the blades of the channel to undergo this lever-like flattening, and then this opens the central pore and allows ions to enter and excite the cells. And Pataputian, Artem Pataputian, he's a bit poetic. He's quite nice with his words. And about this, he had said that what makes touch so unique is that receptors have to transform physical force into a language that cells use to communicate with each other. So these ion channels, these tiny little microscopic ion channels, are a point of entry into this whole system of how we sense touch and how we sense pain and how we sense our blood pressure. So every time you touch your finger or feel something hot or cold, one of these tiny receptors gets activated, and you can think of it like an electrical switch. And a tiny little bit of electricity gets through the sensory neurons, which is enough to tell your brain that something, let's say, has touched the finger on your right hand, right? And to tell you if what you touched was hot or cold. Then to put these laboratory discoveries kind of in a broader context, I mean, that's kind of the cool thing, is when you've discovered something really fundamental and really basic, you see that it's not just in your laboratory setting where it's applicable, but it's something that can support general processes in different species and in different settings. So the TRP family of receptors are molecular detectors of a whole range of stimuli that produce heat and cold and pain. It's not just capsaicin or chili. It's not just for our culinary delight of enjoying spicy food that we have these receptors. So TRP channels, for example, are why a wound keeps hurting long after you've had a bad injury. So if you slam your finger in the door or if you burn yourself, this causes cells to release molecules. As David Julius calls it, an inflammatory soup of different molecules that are released that signal that tissue has been damaged. And so these are the so-called endogenous agonists, or these protons, these that are released from damaged cells. So it's not just capsaicin or heat, but these other molecules can also bind to the TRPV receptor and maintain its level of activity. So after you've had a bad injury, your finger, let's say, if you've burned it, it's very sensitive to heat for the next several days after you've burned it. And that's because these molecules are lingering and they're causing your TRP receptors to remain very, very sensitive. So that even a slight amount of heat becomes very, very unpleasant if you've been burned previously. This is also the case with sunburns. So this is actually really important for our survival and for our sense of self-preservation, because if you're injured, you want to avoid further injury. If some part of your body is hurt, you want to protect it. And these kinds of receptors help facilitate that mechanism. So sensing these kinds of pain is essential for self-preservation and has great survival value. And this has been demonstrated, for example, in the laboratory using genetic experiments. So this is focusing on the TRPM8 receptor. This is the menthol. This is the cold receptor. And this mouse is normal. This one is so-called wild type. It's genetically, it hasn't been modified at all. And it's in this 2-chambered box with a 30-degree side and a 20-degree side. Oops. there are these heating elements and cooling elements that are underneath either side of the box. We'll wait for the video to come back up. So 20 degrees and 30 degrees. And the animal naturally has a tendency to stay in 30 degrees because it's warmer, it's more cozy, it's a more comfortable temperature, and they will avoid the 20-degree side of the box. So then, we'll get to it in a second, they made knockout animals, where they took the gene for this menthol receptor and they knocked it out. So we'll come to it here. The TRPM8, these are the knockout animals. They don't have the gene, they don't have the menthol receptor, and they have no preference for warm or cold, presumably because they don't experience cold. That's kind of the crazy thing. The animal has no preference. It goes back and forth, and it's not until they lower the cool chamber of the box to very, very cold temperatures, like freezing, to 5 degrees or 0, before the animal will start to avoid being in the cold side of the box. And again, this is kind of evident of the survival value that these receptors bring, because you'll get frostbite, you'll freeze to death if you don't know that it's cold. And David Julius's lab has done lots of really beautiful work outside of standard laboratory species. They looked in other heat-sensing animals like vampire bats and snakes, pit vipers that have the heat-sensing organ. And as it turns out, if you look in the heat in the nose of the vampire bat, there's this heat-sensing organ that has a very, very high concentration of nerve endings. And on those nerve endings are TRP receptors. They have a certain variant of the TRP receptor that is a little bit more sensitive to lower temperatures, let's say off the body. And the animal uses this to locate sources of blood in sleeping animals, like a pig or a cow, so that it can locate the blood, bite at the relevant spot, and then have its meal and fly away. And again, not just the receptors used for sensing, but in toxins. You see toxins that cause TRP receptors to open up and stay open, in different venom from different spiders or scorpions and different animals in the animal kingdom. So TRP receptors and their mechanism of causing or alleviating pain are things that you see kind of throughout the animal kingdom. And not only is it expressed in animals, it's expressed in us. And understanding how TRP receptors work has also given inroads into treating chronic pain, like hypersensitivity to temperature or pressure. So again, after having had a sunburn, you are more sensitive to the heat. You'll stay out of the sun, which is good, so that you don't burn yourself further. But in the case of chronic arthritis, this is just where something has kind of gone off the rails. You're having chronic pain. And the TRP receptors have been used as targets for pharmacological agents to go in and block the TRP receptors and reduce the chronic pain that people have. And then to contextualize the piezoreceptor, the touch receptor. These sense mechanical force not just for our sense of fine touch, but it subserves many functions throughout the body, well beyond touch. So this is an X-ray crystallography rendering of the actual piezo channel itself, the piezoreceptor itself. It has this ribbon-type diagram shows that there are lots of so-called transmembrane domains that keep it thoroughly embedded in the tissue that it's in, so that it can sense touch and vibration and these kinds of things. And again, this is the outside, this is the inside, and when the channel opens and ions can flow through, it is a gateway to all manners of sensations. So not just fine touch on our fingertips, but again, it enables this sense of proprioception. So these things are embedded in stretch receptors in our skin, in our muscles. It's all over the body, and it allows us, again, to keep a map of our body position in space. Piezoreceptors also sense stretch in the stomach, so that you can feel at the end of a meal, if you're getting full, that your stomach is stretching. That's because of piezoreceptors. But the thing that was a little bit more eye-opening for me was to realize that the entire body relies on mechanical transduction forces that is essential to all kinds of different functions. So we have piezo1 in neurons that are around our blood vessels. And these act as baroreceptors, or pressure sensors. And it's critical for us to sense and regulate our own blood pressure. It is even expressed within the erythrocytes, the red blood cells, so that they can sense when they're full or empty. Piezoreceptors are also, quite out of control in cancer cells. And as it turns out, piezo-1 mediates the proliferation and migration of different invasive types of cancer. So you could imagine, and I don't know if this is actively being sought after or not, but you could imagine targeting the piezo-1 receptor so that cancer regains its sense of mechanical growth and doesn't grow and doesn't invade into tissue during metastasis the way it normally does. And another example is in the bones, that piezo-1 regulates bone growth and it mediates mechano-stimulated bone homeostasis. So, for example, if you're an MMA fighter and you're constantly hitting and being hit, you need thicker, denser bones, and the piezoreceptors are sensing these hits and triggering your bones to grow more densely. So, with that, I myself am going to stop presenting. And I'll start a video that the Kavli Foundation provided. It's about 6 minutes long, and you can actually hear from the prize winners themselves. They can tell their story of their own discoveries, and they can put their discoveries in context. And they're both really, really excellent communicators. And if you have the chance to hear them speak later this week, I would strongly recommend it. So.
Studying sensory systems is a really unique aspect of biology because, every creature sees the world in a different way. We see the world in a different way than a honeybee or a snake, and that really depends upon the biophysical and molecular sensors that we have in our bodies that interpret all these signals from our environment and then convert this information that our brain can kind of reconstruct as our world.
Over the last few decades, we have learned a lot about how we see, smell, taste. Of course, smelling and tasting is all based on chemical sensing, and this is actually the language that most of our cells in our bodies use to communicate with each other. But the somatosensation, which is sense of touch, pain, proprioception, etc., all of these depend on sensing physical stimuli, which is very different. So how do you sense temperature? How do you sense pressure? And translate this into a language that neurons understand, which is an electrical signal, was not known. It was known that these specialized proteins called ion channels exist, but their identity was not known. So my lab has found ion channels responsible for pressure and earlier work in temperature. What your skin is sensing is the indentation. So anytime you touch, your skin is being indented. What's really fascinating is we say there's the sensor in the skin, but actually most of the sensing is happening at this very, very thin, very long projection of a sensory neuron that starts in the back of your spinal cord. This neuron sends processes everywhere in your body, including the skin. And at the tips of these neuroids, that are sub-micrometer in length, sits this tiny, tiny ion channel, which is in nanometers length, that when the plasma membrane, which is the fatty surrounding of a cell that protects the cell, there's this little protein sitting there. And this protein is, as you can see here, is a molecule that comes with 3 identical members in different colors. They come together and sit in the membrane shown in this green color, and they actually bend the plasma membrane. This piezo-2 protein, there's 2 of them, we call them piezo-1 and 2, are proteins that are encoded by DNA, and so a gene. So there's this gene that its only role is to make a protein that sits in a few cell types on the membrane, and its only job is to sense pressure. And when this cell gets pushed or dented, they flatten. And when this flattens, conformation of the protein changes, and ions start flowing through this protein. And when that happens, you have the first signal that starts the process that we call touch.
What molecules in our body is it that react to heat and cold?
At least as far as we know, most of these molecules belong to the same family of membrane proteins, these ion channels called TRP channels, which are actually first discovered in flies, in fruit flies. But in mammals, a number of them, different ones, and we don't know yet all the ones that contribute to temperature sensation, but at least two of the ones that we've looked at, the receptor for chili peppers, for capsaicin, and the receptor for menthol, these TRPV1 and TRPMA channels, they're sort of molecular cousins in the same gene family, and one of them senses heat and one of them senses cold. This is a model of the capsaicin receptor. This hole in the middle opens up like a donut hole, and it allows ions to flow down their what we call electrochemical gradients. So outside there's a lot of sodium ions, and the sodium ions rush into the cell, and the currency of communication in the nervous system is electricity. So when ions are flowing across the membrane like that, it generates an electrical current, and then this excites the neuron. That initial excitation is then carried along the neuron, say from your finger all the way along into your spinal cord. That information gets relayed to another neuron that carries that ultimately to your brain. Your ability to recognize something hot or cold is determined by the biophysical properties of this protein, which is really kind of a magnificent thing, right? Our route to discovering that was a little bit indirect in that we didn't go in first asking what are the molecules that sense temperature. We asked what are the molecules that sense these pungent agents, these natural products, menthol, first capsaicin and then menthol. And with capsaicin, we identified this channel, and we knew it was going to likely be important for some aspect of pain sensation, but we didn't really know what the endogenous normal activator, physiologic activator might be. So we threw all kinds of things at it that we thought made sense in the context of pain, like neuropeptides, neurotransmitters, and didn't really find anything that seemed to be consequential. And so then we decided, our pain system does more than detect chemicals. It detects physical forces like touch or temperature, and so we tried changing temperature. Cold didn't do anything to the capsaicin receptor, but heat did. And so we realized that that the capsaicin receptor and the heat receptor were the same. And in retrospect, of course, everybody thinks we made the connection right away because you eat a chili pepper. But it didn't have to be that way. It could just be that they're on the same cells and you activate the same pathway. And so that was kind of a eureka moment when we realized that this is kind of a very parsimonious explanation for your everyday experience. Among sensory systems, I always say, I think the somatosensation and pain is probably the one that's most important for our survival and well-being. Because if you don't have it, you really have trouble learning how to avoid things that can be injurious.
Please welcome on stage David Julius and Ardem Pataputian.
That's what's being said in Oslo, like right now. They're welcoming them on the stage and giving them their prize. But they'll be here. They'll be in Trondheim later this week. So, for example, you can see them at the Meet the Laureates event. That's on Thursday at Gløshaugen and Realfagbygget. That's going on from 10 to 2, and David, Julius, and Adam Partaputian specifically will be on from 12:30 to 2. So that's it for me. Thanks for your attention.
Thank you so much for this lecture and the good way that you are able to explain to us, so we at least have a feeling of being able to follow. We are opening now for some questions, if you would like to, or comments on this lecture. Please. Do we have the microphone? Yeah.
Is it on? Mm-hmm. First of all, thank you. Very interesting, very well presented. The TRP sensors, tryptophan, why TRP?
Oh, TRP. It's. I can tell you the name, but I can't tell you something. I can't give you a deeper biophysical explanation. But it's transient receptor vanilloid potential, or transient receptor potential vanilloid. So it's of the a class of receptor called vanilloid receptors. And TRP is just transient receptor potential, meaning I'm guessing if you apply vanilloid, or another compound, that this would lead to a transient opening of the receptor, so it would open and then close. Pharmacology and naming receptors is kind of a funny business because it can be named after the compound that leads to its maximal activation, but not always. So that's why TRIP. It's not related to tryptophan, the amino acid, or no. It's sensing different different classes and compounds of molecules. Yep.
More questions?
If not, that's fine because this is not my. This is what they do. It's not what I do.
I would like to give you one, but on a general base. It's fascinating to hear about this, and obviously the importance of these findings is easy to understand, for it's connected to our survival.
Oh,
But these receptors of pain, for example, it was mentioned arthritis. I'm wondering, can you think of, or are you thinking of that this could be doors opening into new universes for example, treating pains connected, cancer, all injuries, those specifically having pain which no medical today can help for? Is
Right.
that something could be possible?
In a word, yes. So when you have the the example that I showed was for the TRPM knockout mouse, and so that's the menthol receptor. And you knock that out, and the animal doesn't have a sense of cold. But there are, I mean, this work has been going on for tens of years, and they've made different knockout mice that are missing different genes of different classes of the TRP receptors, and the knockout mice lose all kinds of senses of pain. They lose pain related to not just heat or cold, but also specifically due to cancer growth and degeneration that's related to cancer. So I can assure you, I mean, I don't know because it's not my specialty. I can't tell you exactly what agents are targeting which receptors, but it absolutely is being pursued. And again, that's kind of the cool thing when a fundamental new insight is made that subserves all kinds of different functions in the body. You're producing all kinds of new targets then that the pharmaceutical industry, if it could be beneficial, that they can go after and look at and try to either augment or block. And like I said, it's not really my field of study, but I do know from having read up on this that the TRP receptors are major targets that pharmaceutical industries are looking after. Pharmaceutical companies are looking as therapeutic targets. It's not just arthritis. No.
So we say thank you very much for doing all the work to find out this and to present this. Thank you, Jonsson. Then we'll have a 10 minutes break, and then we come to lecture 2 in 10 minutes time.
lovers lie entangled like a river running wide. Never stop to wonder what tomorrow may bring. Kisses are as natural as birds up in the sky. Always there, unaware of darkness to come. I always thought that I would be with her forever. Although we tried, it was a hopeless endeavor. If I could speak, I'd say that we should be together. What will happen on the day the rivers have run dry? Will I still try to feel as we did
Ok, da fortsetter vi. Vinnerne av Kavli-prisen 2022 vant prisen for sin banebrytende oppdagelse av gener, som ligger til grunn for en rekke alvorlige hjernesykdommer. The winners of the Kavli prizes in Neuroscience in 2022 pioneered the discovery of genes underlying a range of serious brain disorders. The lecture is given by Mariam Ziai, Associate Professor at the Kavli Institute for System Neuroscience. The stage is yours. Welcome.
Oh, that's... Hi, everyone. Thank you so much for coming tonight and showing interest in science. As it was mentioned, so I will be presenting the Kavli Prize winners of 2022 for their discovery of genes underlying a range of serious brain disorders. And so our laureates are Professor Jean-Louis Mandel from University of Strasbourg in France, Professor Christopher Walsh from Harvard University in the state, Professor Huda Zogby from Baylor College of Medicine in the state, and Professor Harry Orr from University of Minnesota, again in the state. Just a disclaimer before we go on that this talk by no means going to capture the depth of the knowledge that they produce in the field. It's just going to spark some ideas that for you guys to come and see them on Thursday, as Jonathan mentioned. There's a meet the laureate session on Thursday as well. Okay. So following what Jonathan mentioned, let's think about how different species see the world. Just wanted to give you an example of a shark. So a shark can actually hear its prey from a kilometer away, or can smell its prey more than 100 meters away, can feel its prey when it's approaching. So it can't see the prey, it's just very blind until the prey is really nearby, but can smell and feel and perceive the electrical activity of the prey. I don't understand, like it feels strange to be able to perceive the electrical activity or like electrical kind of signals from people. But yeah, different species perceive the world differently, and us human also perceive the world in different ways. So some of us, we are blind to the colors, so we don't see the full range of colors. Some of us, we can't see the colors. And so what makes this kind of unique feature that we have, like the way that we can see and perceive the world as human? So this is a Google Earth, if you will. Okay. Oh. I'm just going to play it here, see whether it's going to play. Yeah. So our brain is very complex and unique. So this is, as I said, this is a Google Earth from the brain. So we took these pictures from a really high-resolution scanner that we have. And so you can see that coming from the top and then going into details. So we have one of these scanners actually in Trondheim that it's unique in Norway. And we can go deeper and we can have really high resolution of different brain structures. We can also look at how different brain areas are connected together and how different brain areas communicate with each other. This is a very special technique that it was developed in other universities, but then you can go deeper and then you can look at the brain in different layers and different modalities. So you can look at the structure connections, or you can look at the oxygen consumption or blood consumption of the brain. So in a minute, in a second, you will see that this vasculature of the brain and how it's formed. But the brain is never sleeps. So the brain is always working and always trying to combine the information that we receive in our daily life. And even when we are asleep, our brain is still trying to combine this information and get rid of the waste during the night in our brain. So in next slide, I can tell you that the brain, the structure that you just saw from a really high resolution scanner, is made of different cells called neurons. So neurons are the building blocks of our brain. See whether this is gonna play. Yes. So our brain is composed of more than 86 billions of neurons. And these neurons, as you've heard from previous lecture, the electrical activity is the currency in our neurons. So our neurons have these branches that they receive information and they transfer the information to the next one. So these connections that we see here, they're called synapses, that they transfer information, receive information from one cell, and then send it to the other cell. So the combination of these series of activation between different cells make the way that we perceive the world, we see the world, and our brain governs everything we do, our emotions, our thinking, our feeling, and our actions. So these neurons are responsible for the way that we feel and we perceive the world. So these, I will tell you in a minute that the way that these neurons communicate to each other is through a currency or a messenger that is a protein, and our gene regulates this protein. So the way that the information is perceived from one cell and sent to the other cells is through these proteins that I mentioned. So what is the gene? So our gene regulates this production of the protein and transfer of this information from one cell to the other. Our gene, we receive our gene, nearly half of them from our father and nearly half of them from our mother. So this combination of the genes from our parents makes us unique, and everyone in the world makes everyone unique in the diversity that we see in the world. So you can think about the gene as a recipe book. And so you have different genes as different recipes, and each gene has different letter and different ingredients in it. So if the ingredients are high or low, so the product or the food is not going to be as tasty. So the product could be a phenotype, a cake. So I tend to kind of lower the sugar a little bit when I make a cake, but sometimes it's okay. But if it's too low, that's not a tasty cake. So it depends on how much of these kind of ingredients are modulated or modified, then the product or phenotype will be different. So our gene defines our height, our fingerprints, or the color of the eyes. And then they are compacted in a chromosome that, again, we get from our parents. So females, they have 2 XX, and then males, they have 1 X and Y from their parents, and that's the only difference. The rest of the chromosomes are similar between 2 genders. So the genes not only do define this color of the eyes or fingerprints or the height, they also they are important for the communication between neurons, as I mentioned. So the neurons, the proteins that is important for sending signals from one neuron to the other neuron, for the development of the neurons, for growth of the neurons, these genes are really important. So our genes basically are important not only for these physical phenotypes, but also for brain function. But then the genes, they define us, but they don't make us. So there are all environmental factors that impact and they interact with our genes and make us who we are and the way that we respond to the world and perceive the world. So Professor Christopher Walsh was looking into how these neurons communicate and the genes in each of these neurons. So through this process that he was looking into neurons, he found some genes that they were muted. So in a way that when we get our genes from our parents, in every generation there are slight changes between generation to the generation. Around 100 mutations happen between generations. And that mutation could be, as I said, these are the chromosome and there are letters, so it could be deleted or it could be duplicated, so we have more letters or we have less letters. And normally it's fine, it functions fine, and it doesn't have any consequences. But it happens, if it happens in a specific location. It's all about real estate, so it's all about location. So if it happens in a location that matters, then it could cause neurological disorders. And that's the discovery of Professor Walsh and also the rest of the Nobel Kavli Prize laureates this year. So we knew autism spectrum disorder is a genetic disorder, but he found out what is the cause of it and a kind of genetic factor contributing to it and where is located. So you probably know about the autism spectrum disorders, and it's one of the neurodevelopmental disorders that have three main kind of symptoms. So they have difficulties with communication and interaction with others. So the social factors are impacted in a way that they have difficulties understanding and understanding others' feeling and thinking. So we call it theory of mind ability. It's affected in this disorder, or it's a spectrum. So some of them, they might have really difficulties in understanding others. Some of them, they have less difficulties. And they have restricted, they have restricted interest in and also repetitive behavior. So they might repeat words again and again, or they show repetitive movement. And so some of these symptoms might impact their intellectual ability or their functioning in work and life. But as I mentioned, it's a spectrum. Some of them, they are quite severe. They have severe symptoms and some don't. But I don't have time to go through work that's done by Christopher, but the work that he's actually currently doing with others in the field, they established a new center that is looking at the evolution of the genes and evolution of our species in a more systematic way, and looking at the genes that are contributing to the evolution of human brain. So, as I mentioned, there are like these chromosomes, and then some of them, we have X's and Y's. And so Jean-Louis Mandel found that there's a X chromosome that it might be broken, the end of it might be broken, and it's kind of that's where the name of the syndrome, Fragile X syndrome, comes into play. And then that it could cause various symptoms or clinical kind of feature. It's a very rare disease, but so if you think about autism in 1 in 100, 1% or 2% in Europe, Fragile X syndrome could be 1 in 4,000 boys or 1 in 8,000 boys. It depends on the countries as well. So it's much more rare than autism spectrum disorders. But then the symptoms include, like you can see, large ears in kids, especially boys, flat feet, slow muscle tones, and soft skins, so some intellectual disability, some social anxiety, and also inability to focus or hyperactivity. And it's a common genetic causes of autism as well. So you can see that these changes that we see and this mutation we see in the gene could cause this syndrome, rare disease that they discovered and awarded the prize for this year. But I found the story of Huda really exciting and fascinating, so I wanted to tell you a little bit about it. Huda was a clinician, so she was a pediatrician, or she still is a pediatrician. But first she saw she saw one of her patients, Ashley. She was normal until age of 2, and then after that, she started to lose her cognitive ability to speech, and then motor movement was also deteriorating. So she found this was kind of clinical symptoms of the Rett syndrome, and then that was the first patient that she saw, and then she realized that actually she wanted to do more. She doesn't want it to just be a pediatrician, and wanted to understand why this is happening. And she was adamant that this is a genetic disorder. Everyone was like, this is not a genetic, but she was adamant that you can find the genes. And so she started her research on these patients and trying to find out what is the cause of this. So this is one of her patients that she's describing the symptoms, and you can see that. So you can see that she is wringing her hands, and also she's showing rocking movements, so this is kind of symptoms similar to autism. And then when she starts walking, she's very stiff, and the planning for the movement is hard, is also similar to some of the neurodegenerative disorders called Parkinson disorders. And so she's unstable and it's very rigid. So this disease, or Rett syndrome, is sporadic. It's one in a family. It's not heritable. Yeah, and then they are, as I said, they're born normal, and then they lose their cognitive and language function after age of 2 or 3. So after 16 years, it took her 16 years to discover the genes that regulate these proteins and causes this Rett syndrome. So they found this protein called MECP2 that is expressed across the brain. And so you can see this is in 10 weeks old fetus. And then all the way to 10 years old, you can see how this protein is expressed all over the brain. So as you can imagine, that if this amount of this protein is changed, if it's higher or lower in an abnormal way, then it could impact a lot of cognitive functions or a lot of functions in general, because it's expressed across the whole brain. And that is in fact the case. This is kind of a schematic of the mouse model, where you can see that if this protein is expressed in different brain areas, could cause various clinical features or symptoms like seizure, loss of speech, or aggression, difficulty in breathing, and anxiety. And also this is a very that could cause various cognitive deficits. So the level of the protein, if it's high or low, as I mentioned, it could cause various cognitive functions, dysfunctions and disorders. And that's how this discovery kind of led to more discoveries, and that this MECP2 could cause Rett syndrome, intellectual disability, autism, or other like early onset schizophrenia as well. And that's the case for autism. As I mentioned in the work of Christopher Walsh, the discovery of the genes that it was important for autism also led to more discoveries that's finding that actually that gene is responsible or involved in schizophrenia, ADHD, in epilepsy. So this kind of poses a question of whether there are many genes and one disorders, or whether there is one gene and many disorders. The question that we still don't know the answer. And this is kind of the topic that this award opened new questions and opened new kind of ways of discovering how these diseases are impacted by the gene mutations. So the last one, Harry Orr was also collaborating with Huda. So Huda contacted Harry to be able to work on the similar kind of genes that they were working on, because Harry was working on a type of disorders that causes mutation in similar area that Huda was working on, with similar ataxia problem, meaning that they had neurodegenerative disorders, that there was muscle stiffness, as I showed you in the video. So Harry accepted to collaborate with Huda, and then at some point they split the area that they were looking at into half, because it was just a lot of genes that they had to go through. So they split it into half. They started from outside in, and exactly in the same day they discovered the gene that was responsible for the ataxia. The protein is right in the middle. So through the collaboration, after many years, they finally found the protein that was responsible for this disorder, the ataxin-1. But then also Harry's work and also Huda's now, they are kind of, as I mentioned, they're opening new worlds and new doors for curing neurodegenerative disorders, in a way that if this is these genes modulating or modifying this protein, and then the high level, low level of the proteins are important for these clinical symptoms, can we modify them somehow? And so they're working on these genes, but not also on just that disorder, but also AD and PD, so the Alzheimer's disease and disorders and Parkinson disorders, which the level of protein might be different in these patients, and if you can modify it in a optimal way, so you might be able to reverse the symptoms, you might be able to cure it somehow. So they are still working on it, and this is kind of their approach to see whether they can modify these proteins expressed by these genes in other neurodegenerative disorders such as Alzheimer and Parkinson as well. And yes, there are hopes, there are some advances in this area. So identifying these genes that might be responsible for rare disorders like Fragile X syndromes, for example. We have now some ways of monitoring and screening the genes. So when the kids, like in the fetus, that you can screen them and you can identify the genes that are muted. But also from the therapy point of view, or from the therapeutic kind of point of view, they developed some mouse models, as you saw in the previous lecture as well. So they were healthy. This is the kind of a brain activation of a healthy mice. And then this is activation or the kind of a seizure frequency of a Rett syndrome mice, which you can see that there's just very frequent seizures and electrical activity here. But when you modify the protein in a way that is optimal, then you can reduce the seizure frequency in these mice. So there's a hope in a way that you can modify these proteins and the expression of these genes, so then there's a way that you can reverse the symptoms in these models, animal models. And then the other model was the fruit fly model, that this is eye cells, and then you can see that the cells are quite distinct and very healthy looking, and then when they have this ataxia and neurodegenerative disorders, it looks different. And when you modify the protein in an optimal way, you can kind of reverse back and you can have a bit more close to the healthy cells as well. So it depends on how they are working on modifying these proteins that could lead to reversing the symptoms. Okay, so that's it for me, and I just wanted to show you the video that laureates talk about their discoveries as well. So it's about 6 minutes, and yeah, let's see what
The brain requires a lot of genes to get put together. We have about 20,000 genes in total in our genome, and the brain uses almost half of them. And so that creates a lot of potential problems, because if almost any one of those genes is not working properly, we can develop disorders of the brain like intellectual problems or cognitive problems.
Our intellectual cognitive abilities, language, writing, speech, is much different than our non-human primate ancestors. And that's due to changes in the DNA that then impact the function and complexity of the brain. But along with that are changes in DNA that causes diseases.
There are many types of mutations, and we have a long way to understand the effect of most of them. We've had better success in identifying the mutations that impair genes, because that's obvious. You've got someone who suffers from a disease, and you study their DNA and you found a mutation, you can relate that to the cause. But we have a long way to identify as many mutations as possible that actually might confer protection and might improve health.
Function of the ataxin-1 gene, its primary function is to encode and give the genetic information for the ataxin-1 protein. And when we talk about function of the gene, what we're really talking about in genes like ataxin-1 is we're talking about the function of the protein. And that's one of the major questions that we have tried to address over the years, is because we think the function, the normal function, if you will, of the ataxin-1 protein is very much linked to the molecular aspects of why the mutation in that protein then causes the disease, spinocerebellar ataxia type 1. And what we know so far is that an important function of the ataxin-1 protein is to regulate gene expression in a variety of cell types, not just the brain, but also some of the muscle and other tissues in the body. And what we're trying to understand is why certain neurons are more susceptible to the mutation that disrupts that function or alters that function than other cells. And that's an interesting biological question, but also perhaps in the root of answering that question might give us better information of how to counteract the negative aspects of the disease-causing mutation in the gene/protein. But at this time, we're focusing on the nucleus of the cells and regulation of gene expression in terms of its function.
In 2003, the Human Genome Project determined that each human has 6 billion letters in its DNA, 3 billion base pairs inherited from their parents.
Each time we conceive a child, out of the 6 billion letters, there are actually about 60 that will change from parents to the child. Most of them have no effect. Some may have very mild effects, good or bad, and rarely, but not so rarely, it can lead to a dysfunction in a gene that is very important for the brain function. Many of them are not transmitted. Some of them are transmitted. Fragile X can be transmitted in a pretty bizarre way. But because there are new mutations happening at each generation, each time we conceive a new human, then it will stay this background of people with such mutation. And this is something we cannot really prevent unless the only way would be to sequence in every fetus, and then not keep those who have mutations that we can identify as very bad. But this would pose both ethical questions, because we don't always understand what a mutation effect is, and of course for the moment it's not feasible or desirable.
I think one interesting area that we're aiming for is to try to understand how the development of our brain predisposes us to various disorders that might develop much later in life. So at the moment, we know enough to know that the development of the brain can predispose to certain conditions that affect a child, like focal epilepsy or intellectual disability or autism spectrum disorders. We also know that there are roles for genes in creating other disorders that present late in life, like degenerative conditions or Alzheimer's disease. What we don't yet know, and I think we might understand more about soon, is how the pattern of development also contributes to what happens later in life. Not just the genes, but in fact these exposures and so forth that can also alter our genome in a way that happens independent of what we inherit.
I work on childhood neurodevelopmental disorders. In particular, one such disorder is called Rett syndrome. And that disease shows up much earlier in life. It typically shows up in the second year of life, where the babies lose the skills they used to do and cannot speak and cannot learn new things and will not use their hands anymore. They will wring their hands and they have balance problem. But it's because of changes in the function of the brain early on. That disease is very different from the ataxia because it's only one in a family. The ataxia, I told you, it passed from generation to generation, so you have many affected individuals. In the case of Rett syndrome, there'll be a couple who will have a child who's healthy, and then the next child may have Rett syndrome. Or maybe another couple, their first child will have Rett syndrome, but then they have other healthy children. So it's just one in a family, but it's sporadic. It's not inherited. Parents are healthy, child is affected, because in the sperm, in one sperm, out of the many, many healthy sperms that father carries, one sperm will have a mutation. And if that sperm gets fertilized and becomes an embryo and then a child, that will bring on Rett syndrome. So this is a class of disorders which are genetic, but not inherited. So it's still caused by a gene, but it's a random mutation that happens.
Please welcome on stage Huda Zogby, Harry Orr, Jean-Louis Mandel, and Christopher Walsh.
Yes, that's it for us. Thank you so much for showing interest in science again and coming tonight. Yeah, I can take some questions and see whether I can respond to them or answer them.
Mariam, thank you so much for this presentation. I'm sure that everyone here, it's fascinating, the trip you are opening doors and opening doors into a universe that is really hidden. But we of course heard of these genes, but what is it really, and how do they work?
Exactly.
And so I'm really to look into this universe. It's really, really interesting. Is anyone wanting to give a question or a comment or anything to Mariam?
Can't see from here, but
Okay, Jonathan, please.
Based on the work of any of the laureates, are they making progress or inroads on treatments, or are there different avenues of treatments that any one of the investigators is particularly pursuing to treat or reduce the disorder?
yeah, they're actually like Huda and Harry, at least based on my understanding and my readings, that they are using some of collaborating with some pharmaceutical companies, so then they can use to modify these proteins, as I mentioned, using different drugs to bring it down, and then see whether you can reduce the symptoms or kind of symptoms of ataxia, for example. In the case of Rett syndrome, I think she's also looking into some pharmaceutical intervention as well. But yes, they are. And so the impact of this work, I think the fascinating aspect was that it's not only just a rare disease that they, like 1 in, 1,000 or 10,000 happens, and it's just, it's so rare that you might not have any implication, but then understanding the mechanisms of how these kind of rare genes work, and then how it could lead to different dysfunction in the protein and all of these kind of brain disorders, led to more discoveries of that these genes are actually involved in schizophrenia or other intellectual disability or neurodegenerative disorders like Parkinson and Alzheimer's. So that was kind of a very fascinating aspect for me, that how these kind of rare disorders of Rett syndrome or ataxia actually led to more discoveries in other fields that are more common, like neurodegenerative disorders.
Okay, no more hands. I would say thank you so much for the work you have done and shared with us tonight. And in this field, we will for sure read and learn more about in the decades to come. Thank you.
Thank you.